Good morning to all. Once again, I welcome our guest lecturer, Dr. C. Kannan, who has come from Indian Institute of Rice Research, Hyderabad, who has accepted our invitation and going to deliver a lecture on role of microbial secondary metabolites in the management of plant diseases. So let us listen from Dr. C. Kannan.
So, so thanks for the remaining students who are sitting here, and let us excuse the other ones who have left. So actually, this is a very good initiative from TNAU just to make the external examiners give a guest lecture, because the primary role of this is to expose the students to people working all over, because this otherwise you have to go to our sites which may not be of much interesting to you. So when we come here we can explain better what we are doing. So let me just introduce myself from the institute background. So I am Kannan, working in the Indian Institute of Rice Research, which is a component of Indian Council of Agriculture Research. So like ICR has many component institutes, both from agricultural crops, then horticulture, then veterinary, and even dairy is a special institute is there. So we have different component institutes working with a mandate of particular crops, or sometimes bigger institutes like horticultural IH or Bangalore, where there will be multiple vegetable horticulture crops will be there. So I come from Indian Institute of Rice Research. The genesis of this institute is actually we started as a All India Coordinated Research Coordinating Center. So we started as a AACRP Coordinating Center. Then it was upgraded to Directorate of Research, Rice Research, where certain lead research was taken for consideration by the scientists present there, and then afterwards it was upgraded to the Indian Institute levels. This DRR, I mean the Directorate, NRs, National Research Centers, then Indian Institutes, all these are different category institutes in ICAR, where they depend upon certain factors like the number of strength of the scientists, then the mandate of the institute, and then the budget of the institute, of course. So ours is a national institute now. It is one of the premier institutes where we are almost some 82 scientists working on different aspects of rice production, and we are headed by a director, and we come under the crop production division of the ICAR. So why I am telling you all these things is like whenever I speak to my senior and my very good friend Dr. Johnson, so the number of students coming from TNAU to ARS is going down year by year. That is very bad actually, I am telling you. So actually come out of this cocoon of TNA. Of course, it is very comfortable. I know it's very comfortable staying in TNAU. You normally complete PhD, all from UG to PhD here, and then complete your 5 6 years SRF, and then join as assistant professor and continue the same place. So one thing is you lack exposure in different places. Actually, traveling around is very good. So my main aim of accepting this lecture is to motivate you guys for coming up to this ARS preparation. I think there is one ARS. How many of you are in third year PhD about to submit your thesis? Anybody about to submit? Because I think there is a ARS call for us come. I think only now from now onwards it's only PhD. So I think Lokeshwari is so tell her to do well. Let her take some of our help who are all there in other institutes. At least we can just share our experience. How? Huh? Okay. So because that is very important, or at least come for even nobody is applying to any SRF post elsewhere. Hyderabad is one such position and place where it's very comfortable living. It will be just like Coimbatore. Even the climate wise also, only 3 months it is horrible there. Anyway, it is just from the point of exposure only I am telling. Nothing is big, nothing. I am not telling ICR is bigger or the universities are less. And I am not telling in that angle. It is just in the sense of or to reduce your inbreeding depression. Actually, even ICR brought this MSc to some other institutes, some other universities. Then only you are eligible for thing here, fellowships. But now again that is also stopped because university is giving you fellowship. So even that won't happen nowadays. Anyway, so it is just my suggestion. So I think, so coming to this topic today, this is the area where I am working actually. So I work on biological control since my MSc PhD. I am working on biological control, and it was fortunate for me to continue in that same field wherever I go in all the three institutes. I worked in cocoa in CPCRI, and then I started working on tomato and mustard in Jabalpur. And then I started working in rice. But wherever I go, I make it a point that I work on biological control. So after coming to biological control, then when I came to rice research, I happened to have some very good institutes nearby, especially the IICT, the IITs, where we got some of our colleagues who are doing wonderful work on their area of this, especially the metabolites. I mean, they were doing it for some other purposes. Then I thought, like, why don't we work on the metabolite area of biological control? Because biological control means just isolating the organism, making some formulation and applying on it. So there are thousands of formulations of trichoderma. It is available, but what new can we do? So I just thought of working in this area. So naturally, microbial metabolites are going to be the future in agriculture, in medicine, in any of the areas. Metabolites are going to be the future because they provide what to say, undiminishing source of chemicals. In any field, you take microbial metabolite, it can function as anything. It can function in human medicine, it can function in veterinary medicine, in agriculture, in even nowadays metabolites are coming in electronics also. So that way, so I thought, like, let me start something with primarily focusing on agriculture purposes. Then if we can extrapolate it to other things. So basically, what are metabolites? So metabolites are two ways. One is primary and the secondary one. The primary includes most of the essential compounds, even vitamins, your hormones. All these come under the primary metabolites, which are basically required for the functioning of the organism. And the secondary metabolites are, they are not life-saving or life purpose compounds, but they are very important in maintaining the population of the organism. So they help the organism sustain unfavorable conditions, remove the competition, then make their own population domain. So these things help. These are helped by the metabolites which are produced by them. So these are called as secondary metabolites. So secondary metabolites are that way important for us because one thing is they are mostly secreted extracellularly. Most of the secondary metabolites are secreted extracellularly. So one way it is easy to isolate them, it is easy to extract them from the compound from the organism, and then they provide a very good mimic of what we need actually. So from that we can develop so many other compounds. So classification, this is a very preliminary or one-sided classification where this classification depends on the nature of the compound. So we can classify these metabolites into so many different ways, depending upon their purpose, depending upon their chemical, depending upon their structure, so many classifications. So I have given a reasonable classification based on our purpose in agriculture actually. So when the purpose differs, then the classification also differs. So, what are the functions of the secondary metabolites? As I said, they have wide purpose in many of the whatever you name it, they have a role in that. Say, for example, the first thing is they help the organism defend against the enemies and tide over the unfavorable conditions. Then they help in the transportation of metals. Say, for example, iron. Cetrophore is a very common metabolite which is produced. So then they are essentially linked to the symbiotic associationship with many of the organisms, with many of the higher or lower organisms which the particular organism is associated. Then they are very helpful in the reproductive stages of the organism. And then and the main interesting component for our purpose is antimicrobial. This is what we are working as in our group there. So we work on the antimicrobial properties of these secondary metabolites, which can be safely used for the biocontrol purpose. So there are some advantages why metabolite is used in the place of the organism, because when we use an organism, most of us know, like their survival becomes a problem, their handling becomes a problem. We are not able to increase their shelf life over a certain, and then even their delivery method makes a issue. But here, when we convert them into chemical compounds, then it is as good as applying any fungicide, because fungicides have that advantage. You can mix it with any other compound, you can apply it in any easier way. So that advantages which the fungicides have will be there with the metabolites, minus the disadvantages of the compounds. I mean the fungicides. So, how are this secondary metabolites controlled? The secondary metabolites are basically controlled by the same mechanism as like any one of our other external mechanisms. So, the genetics play a very important role. So, these are the classification. Actually, this we have taken it from our Pseudomonas putida. This is the Pseudomonas putida is one organism which we have in our candy. So these are the antimicrobial mechanisms present in from the putida. I mean, our particular isolate, that is PIK1, we have isolated, and we have characterized this. We have just genotyped. This is nothing but transcriptomics which we have done. Of course, many of these works we outsource it, or if our bioinformatician has some time, then we get it done in our own institute. Otherwise, there are very good companies where you can get these services outsourced. No more we have to struggle for our all these type genotyping. And then this is again another one important trichoderma, that is Trichoderma asperellum. And this type 1 is our own isolate. So we have thoroughly studied this organism for many of our purposes. And we have whole genome sequenced it. We have genotyped totally. We have transcriptome studied this. And we have classified the metabolomic classification of, I mean, analysis is given here. This is the metabolomic analysis. So we have got very good class of metabolites from this aspergillum isolate. So we have already developed a very good formulation, of course, with the private company collaboration. We have developed a lipid-based bioformulation for this particular organism, both liquid and solid. So this is going up very well. Next year, we will be commercializing this. Actually, we have already applied for, and we are going to register this as a biocontrol agent, not as only as a growth promoter. Normally, biocontrol agents are registered as growth promoters. So we are going to register this as a biocontrol agent only. So we need certain toxicology studies which we are doing, of course, with the help of our one partner company. So, so when we were looking at the metabolite class of this trichoderma, then this was having very good antimicrobial activity. So we thought we'll extract, mean we purify whatever compound we are getting, which is very active in the bioassays. So we tested, mean we started extracting the compounds. This is how we extracted. It's general solvent extraction process. So when we took the crude metabolite and we did the bioassay against many of our organisms, so they were working very nicely. Then we did a small TLC to differentiate the fractions. So how many fractions are there? Then we could get around 4 to 5 fractions. Then when we tested this individual fractions against the particular pathogens, and then one particular fraction was working very nicely against that most of the rice pathogens. So when we try to purify that particular fraction, we could get this particular compound, that is 6-pentyl 2H pyrone. This is this is one of the most dominant compound when we obtained. This is the proton NMR which we did. So all these things we didn't do it in our own lab. We got it. I mean, we have a collaboration with the IIT Kanpur, so where we could do this most of the NMRs. This is 500 hertz NMR. So this is a carbon NMR, proton NMR. So we found out the structure of this particular compound, and this is a 6-pentyl 2H pyrone one. Of course, this compound is the most dominant in our organism, but when we saw the literature, this compound was already isolated by one institute in Japan actually. So they were applying this particular compound against the false mate disease. But not only that, our surprise, this compound was also used as a food addictive actually, because this is having very good coconut aroma. So this is also used as food additive in Korea. So that is how I said no. The secondary metabolites have wide varied activities. So we tried to purify this. Then we did all the possible research which we can do with this compound, starting from their bioassay, their stability under different conditions. We simulated different conditions in which rice is grown, mostly in the saline, alkaline, drought conditions, semi-arid, irrigated conditions. So we simulated all the conditions, and we worked on this compound. So this compound is coming up in a very nice way. So this is the pentyl pyrone. I have just given some of the properties of this. So there are two different isomers in this. One is 2 alpha pyrone and 4 beta pyrone. So both these pyrones are very good. But our active pyrone is the alpha pyrone, which is used here. So alpha beta depends on the this position, particular position of the oxygen atoms in the either this way or that way. So this isomers give has some differences in their stability. Otherwise, both the isomers were very good, but their the stability of these two pyromers are little different. So we chose the 2 alpha pyrone, which is quite stable and quite bioactive against many of the pathogens. So, what is the next step now? Now, this is how I would generalize whenever somebody is planning to work on metabolites. So, what we do is we isolate a compound, we extract a compound, we see the bioactivity of that, then we go to purify that compound. So, after purifying, identifying their structures, molecular structures, what then? You cannot always culture the organisms and extract the compounds. That is not going to be economical. So what we have to do is we have to find out a way how to synthesize that compound. So this we started working on the synthesis of these alpha pyrones. So this synthesis was very simple because it's a very simple compound. We could do it in 3 to 4 simple steps. Uh, so we have standardized the synthesis of this also, but only problem is now we are using palladium as a catalyst. We need a metal catalyst to synthesize these organic structures. But normally, either platinum or gold or palladium is used. These are heavy metals which can be, but we are looking for some other compound because palladium is very costly. So we are looking for some other compound which can reduce the cost of the synthesis. So this is the synthetic process which is done. And then the other approach will be to, this is of course I have given the mode of action of 2H pentopyrrole. So it mostly inhibits the mycelium, and it creates metabolomic hemostasis. That is, it disturbs the metabolomic pathway of the target pathogens. Then it is restricting the mitochondrial enzyme synthesis also. And even this was found to reduce the toxin production by the compost. This is how it is very effective against the false mud pathogen. You know, it is Islidin is a very important toxin produced by euvirans, which is having a very important role in the symptom development of false mud. So this particular metabolite reduces even the toxin production. When we did it in co-culture assays in the tubes, so we could find out the the quality and quantity of toxin produced by U irons was very much drastically reduced in presence of this metabolite, even at very minimum concentration of 50 ppm level. So this is our studies on the, I think we have done it on two pathogens, that is sclerotium hydrophyllum, and then this is only on stem rot and false met. So we could get very good suppression of both of these pathogens. So then this is one approach of all, like what I said is you start synthesizing the compound. Then the other approach will be like you can find n number of derivatives of this. So that is one important step where you get the structure of a compound. See, in basic words, we see, for example, we uh, we arrived at the structure of aeroplanes using the birds. Okay, but then what we can do? We have to keep on further refining the process of how the aeroplanes play. You know, like the bombers are different, then the passenger jets are different, then the cruisers are different. So like that, here also we got the structure of a particular compound, which is very good in its bioefficacy. Then we can derive so many derivatives of the abscess. Just it is, these are all chemist's role. Like, of course, I am explaining, so don't think like we will be master in all those things. To be very frank, after they did, I learned what they did. But otherwise, it is the role of the chemist. So one thing we have to understand is we cannot do everything. Okay. So our role will be to identify the particular person who can do their job in whatever area we require. So that is how we find collaborators. So this particular gentleman from IIT Chemistry, he was very interested in producing natural compounds. Actually, but his work is on reducing tumors, actually, tumor inducing metabolites from the gut microbiota. So he was working on that particular area. So when I contacted him, why don't you study our organisms also? So he was very interested to get this, because I'll tell you the next slide where we are collaborating with them in this their studies also. So this is how they were producing the different derivatives of the pyrone compound. So this will give you lot of interesting results, because now this is one pyrone which we synthesize, nitroprione. So this will have totally different role, or maybe it can have much better efficacy than our original compound also. So we have to do. So this will be an interesting area. There is no end for it. You can put one copper inside, you can put one sulfur inside to change the entire property. Maybe the stability of the things can change. All those things we can do. Then I said, like, we are formulating our compound. So this is one formulation which we did. It's a biodegradable carbonide metabolite. Metabolite is our pyrone here, nanoparticle. So this we did. What is the purpose of this formulation? Is this actually this our metabolite compound was heat sensitive? Actually, it was around 60 degrees Celsius. It was losing its efficacy. Then we thought, like, how to improve its thermal stability. So we then formulated using bionanopolymer, because that is a patented one. Actually, we will be getting patent this month. Actually, it's all the preliminary. It's going on for 1.5 years now. So all the work is done. So we have been given a provisional patent also. So our final patent is coming in this week. So the bionanopolymer is a microwave digested process. This total process is we did it in microwave digesting. So it was helping the compound tide over even 150 degrees Celsius. So that is how this formulation is protecting the our pyrone. So this is one way of, like, how we can improvise our compounds. So that is what I'm telling. Once you find one particular compound also, it will be very interesting to do so many things with our because there are so many opportunities. It's not like you find a compound, you apply it and then forget it. You can develop it, you can protect it, you can increase the stability, you can increase its form, so many things we can do. Only thing is you need the imagination for that. So this is how my work is still continuing. So my work is next. We are working on the derivatives of that, and simultaneously we are working on other metabolite compounds also. So we will be continuously taking the secondary metabolites from different organisms. I mean, whatever organism we have characterized and we have submitted for in our registry. So we will be working on different compounds from those organisms. So meanwhile, as I said, na, when we go to somebody else who are working on different areas, then we should also give something to them. So this is how I collaborate with others. So this is like we are working in this metabolite compounds because metabolites are metabolites only. It is not from our fungus or their fungi or our bacteria or their fungi. They are also looking from the same. So the main important issues in medical fields now are they are getting AMR, that is antimicrobial resistant strains, which is a very important problem in the medicinal field now because people are illogically consuming antibiotics, whatever, whenever it goes, immediately they'll over the counter they get the antibiotics. So this is becoming a main problem. And not only by consumption, even our disposal of antibiotics. See, what we do is all expired medicines, we just throw it in the drain actually. So they all are important reasons for developing AMRs. So we are working on finding out solutions for this AMR because ours is a soil ecosphere bacteria, so fungi, both Trichoderma and Bacillus, which we have, and Pseudomonas. So they are totally from a different. So if you see the epigenetics also, there, even though the Pseudomonas I mean, Putida from a soil and Pseudomonas from a different atmosphere will have totally different chemical composition. That is how epigenetics works. So we are giving those organisms to them. That is how they are interested in giving something to us also. So, so we are now basically we have these two important. One is the hemostasis in dental. Hemostasis is nothing but reducing your or stopping your profuse bleeding actually. So whenever they conduct some dental procedures, they have a lot of bleeding issues. So now they are applying some other chemicals actually, which are toxic actually to some of the human races, even particular we can say, because they are allergic to some people also. So they are working on some natural products. So that is how our compounds come into picture. And then this AMR. These two are the very important areas which one. And then again, this is the actually the gut microbiome. We know like all our intestines have a huge population of bacteria and mainly bacteria that help in digestion. But not only that, their metabolites have important role in leading to this colon and rectal cancers. So we are working on, I mean, it's mostly they are working, but I'm just assisting them with my organism. They will ask me which particular organism will have which particular class of my compound. So then, because we have metabolomic analyzed our compound. So if at all any class is there, then we'll give our organism, we'll extract the compounds and then give it to them. They will purify it and then do the biopsy. So that is how they are doing it. So things are going on like this. This is what I wanted to just share with you. Any questions? It's welcome. Just it will be a discussion, nothing questions or answers. I may not know answers for many of your things.